Hello everyone, my name is Clément Bouquet. I am business engineer and agronomist at Dorian in Nice, south of France. So thank you all for joining us from all over the world. As you already know, Dorian is expert of data management in vegetal research and editor of the R&D Information System Suite R&D Experience. We are pleased to serve private and public research teams between 5 to 500 persons, notably seed companies, technical institutes, and agro-food industries worldwide. So please don't hesitate to use the chat to ask questions. I will take a few minutes at the end of my speech to answer them. As you may know, today we will discuss on the statistics in the vegetal R&D world. So today, along this webinar, I will first quickly introduce the use of statistics in the plant breeding world, and secondly, talk about some important statistical tools that could considerably help you in your R&D processes. Among others, GxE and SCA, designs, ANOVA, GxE Matrix, interactive graphics, and finally, artificial intelligence. So based on that, I will expose you how to rely on your results along your experimentation process and how to reconcile you with this abstract world thanks to the use of efficient and standard statistics tools as we propose in our software R&D experience. But before to start this presentation, I would like to know which part of this webinar seems the most interesting for you. On the right, you have a tab named Poll, and you can click on it and select your responses. We will check the results together later during the presentation. So let's begin with a small introduction. Why statistics are so important in our life? Statistics play a vital role in every field of human activity. Statistics are used to analyze what is happening in the world around us. As we all know, we live in the information age, full of internet around, where every single activity can be monitored. However, the reality of statistics is a bit more contrasted. Let's have a look on this quote. There are three kinds of lies: lies, damn lies, and statistics. I'm sure you have heard about this expression, which was spread by Benjamin Disraeli, British Prime Minister. This striking phrase contests the reputation of statistics. The implication is that statistics can bolster a weak argument, or that statistics can be used to prove anything. As an example, this ironic graphic below tries to find an opportune and of course false correlation between the number of people who drowned by falling into a pool with the films where Nicolas Cage appeared in. Of course, it doesn't make any sense. However, it reminds us a basic and fundamental principle: correlation is not causality. There are many ways to produce misleading conclusions. The first way is linked to biased samples. A non-random sample can base the result from the beginning, as we have seen with the example. But we have also overgeneralization. The results from one population may not apply to another population. We also have false causality, because correlation is not causality. Incorrect analysis choice, and finally, data manipulation. It is a serious issue in the most honest of statistical analysis. Outliers, missing data, and non-normality can all adversely affect the validity of statistical analysis. Statistics respect the rules of the three C's: communication, credibility, and convergence. The communication is expressing or communicating your results in numbers to convey your findings. It's much better than using terms like almost as large as or slightly greater than. The second one is the credibility. The use of numbers also provides credibility to scientific studies. Use of statistics requires statements about experimental designs, methods, and database structure. And finally, convergence on truth. But what is truth? When it comes to scientific research, a goal of our work is to converge on principles that inform us about how nature operates. Numbers and statistics help us tighten our confidence in the results. Romain, just before the next slide, can we check the results of the poll? What do we have?
Yes, sure. So we've asked everyone what's most interesting in this presentation, and 27% of you, nearly 30, replied ANOVA and GxE Matrix.
Okay.
And then there's the parental choice with GCA, STA, equality with experimental designs.
Interesting. Yeah, ANOVA it's a tricky key step for the statistics in the breeding pipeline. So maybe I will focus a bit more on this part during my presentation in order to help you managing this type of statistics. Let's continue. First of all, let's begin with the concept of combining ability. Identification of the best performing lines and identification of the lines which can be used as parents in future crosses are two principal objects considered in most cord breeding programs. The best performing lines are selected based on conducting multi-environmental trial following statistical analysis. For the breeder, this step is one of the most critical challenges of all the breeding process. Fortunately, some tools like general combining ability and specific combining ability can help the researcher in his decision. So let's try to define and understand this concept. Combining ability is defined as the parent's ability to combine among each other during hybridization process such that desirable genes or characters are transmitted to their progenies. The general combining ability is the average value of the inbred line based on its behavior in crosses with other lines, whereas the specific combining ability is defined as the value of the line in crossing in a specific cross. From a statistical point of view, the GCA is a main effect and the SCA is an interaction effect. The two main uses of these powerful tools are first, the identification of the best performing lines, of course, and the second one is the selection of the lines which can be used as parents in future crosses. So although these two concepts can be useful for the breeders, we don't have to forget that GCA and SCA values are relative to a particular set of inbred lines included in the hybrid center test. Okay. General and specific combining abilities are complementary and they produce different effects. As a high GCA highlights additive effects between genes, a high SCA is regarded as an indication of lossy with dominance and epistatic interactions. Also, as a high GCA is related to homozygosity, a high SCA is more related to heterozygosity. And as a result, high GCA produce a high breeding value, often seen on autogamous species, whereas high SCA produce a high heterosis, often seen on heterogamous species. And an important thing also is that only GCA is used for selection and is heritable and fixable. So as you can see in this picture, general and specific combining ability matrix can be easily created on RNDX. Researchers can choose between DLL or factorial calculus depending on the characteristics of the crossing materials. These matrices are highly configurable, dynamic, and a quick filter is available to optimize results display. So to resume this first part, GCA and SCA are two complementary tools for performant line selection. Another fundamental step in the breeding process, where statistics play a critical role, is the choice of the experimental design. These designs have a high impact on the conclusions that could be drawn, and some materials with bad characteristics could be selected due to irrelevant experimental design choice. That is why we will review some of the most famous designs used in plant selection with their respective characteristics. So first of all, let's define the concept of a design. It is a type of plant arrangement which is used to a set of materials or treatment in order to draw valid conclusion. A design has three basic principles to respect. The first one is the replication, that is the repetition of the material or treatment to provide an estimate of experimental error. The second one is the randomization, that corresponds to the allocation of the materials or treatments to the different experimental units by a random process. This randomization will have a big impact on the accuracy of the results. And finally, the local control, that is the principle of making use of greater homogeneity in groups of experimental units for reducing experimental error. For example, local control will be different if you have a factor variable in strips or in patches on your field. Three main purposes of choosing an appropriate experimental designs are the estimation of various statistical estimates, testing the significance of difference among various subjects, and finally the proper interpretation of scientific results in order to draw valid conclusions. The standard version of our software ANOVA proposes 11 experimental designs with automatic randomization already configured for your experiments. These designs can be used in several experiments in different locations. Other specific designs could also be easily created by the user depending on their needs. So today in this webinar, I will present you 4 designs that you probably already use for your experiment. The completely randomized design, the randomized complete block, the Latinized alpha design, and finally the split plot design. But do you really know what are their respective pros and cons, and in which condition use one design instead of another? So let's have a look on it. So the completely randomized design is used when the experimental unit is homogeneous. So this design is especially used for plot culture experiments. The principle of local control is not adopted in this design because homogeneous blocks are not found in this design. The second one is the randomized complete block, which is the most commonly used design in plant testing. This design is useful where the factor variation between the blocks is significant. The principle of local control is adopted in this design because the experimental field is divided into homogeneous blocks. This design can test up to 20 treatments. Beyond 20, the efficiency of error control is decreased due to increase in heterogeneity within the block. As you can see in the picture, there are some problems of variety repartition, for example for the variety 5, 7, and 12. The Latinized alpha block is a double system block where materials are randomized in rows but also in columns. It's not really known in the plant breeding world, but it's one of the most efficient designs for local control. Block heterogeneity are well managed as double gradients. The only disadvantage of this type of design is that it's a bit hard to implement and highly depends on the number of materials that you need to test. The final one is the split plot design. This is the most famous design for multi-factor studying. In this design, several factors are studied simultaneously with different levels of precision. It includes factors like irrigation, sowing dates, seed rates, and many other ones. The layout and analysis are more complicated than for other designs. Also, the main factor, which is studied in large plots, is analyzed with less accuracy than the other factors. So as we have seen, the choice of experimental designs have a huge impact on the accuracy of the results, notably on the analysis of variance, most commonly named ANOVA. But I have a question that I really would like to ask you. What are the main issues that prevent you from performing reliable statistics? So for example, is it the place available? Is it the number of seeds that you have? Or even your knowledge of statistics? So please share it with us in this poll, and I will respond it, and I will analyze the results a bit later. So let's continue with ANOVA. ANOVA is a statistical tool that has several applications in the plant breeding context. It is mainly used to test for differences between treatments in an experiment. Common examples of treatments are genotype, location, or even variety. In the software R&D experience, every user can easily load a complete statistic report containing all the analysis of variances required, based on the parameters set previously. The ANOVA can be done on several variables at the same time, with raw, estimated, or adjusted data. To fully understand group differences in this ANOVA, multiple comparison analysis is added to the test to test the difference between particular pairs of experimental and control groups. So several methods are available on RNDX, like LSD, Newman and Kulls, Tukey HSD, Dunets, and Dukan. As you can see on this video, we firstly see the graphic with the materials and their results. So the checks of these experiments are in green to distinguish them from the others. Below, there are the ANOVA results with the different sources of variation. That could be the materials, the replication, or the location, for example. Then the free residue analysis for subjective validation. And below, a feed map with residuals in different levels of gray, allowing to distinguish extreme materials. And finally, you will see a table with material ranking according to their results and multiple comparison analysis with group creation. So in order to add more accuracy on the ANOVA, more and more researchers are considering the influence of gene-environment interaction in their statistical analysis. The GxE interaction could be defined as a differential in the responses of two different genotypes to environmental variation. Let's have an example. A variety A will respond positively to a specific environment, whereas a variety B will respond negatively but to the same environment. And these differences of response are named gene-environment interaction. And so this GxE can be seen as a new selection criterion for variety-specific adaptation to environments. The software RNDX provides a powerful matrix tool for GxE interaction analysis. Values in red traduce negative interaction, as values in green traduce positive interaction. Results can be sorted and filtered for easy material classification and comparison. As an example, we can also imagine the combination of this GxE matrix to a dendrogram to create location and variety clusters according to their common profiles. RNDX includes also many interactive graphics, such as scatter plots, box plots, PSA analysis, and histograms, among others, that are very handy and useful for analysis. So material plotted can be selected to be found on their respective position in the table nearby. This functionality produces an easier results visualization and a gain of time for the users. These graphics could be associated with other tables and genealogy graphics in link presentation to study the linkage between phenotypic and genotypic data, for example. So Romain, before to continue, let's check the results of the poll.
Yes, sure. So we've asked what prevents you from doing reliable statistics. So a large majority of you said lack of knowledge.
Mm-hmm.
Here we are to discuss it. Glad to add something with you. Then you have a lack of space in the nursery.
Okay.
And then lack of reliable statistical tools.
Understand. Interesting. Very interesting. Yeah, lack of knowledge, I think it could be a big issue. And that's why we are doing this webinar today with you, in order to give you more tools and more ideas to do well your statistics in your day-to-day activities. So we are pleased to help you improving these skills. So last but not least, I also wanted to talk about artificial intelligence in vegetal R&D. So at Dorian, we trust in this new technological area to solve your breeding issues and help you in your decision-making process all along your breeding pipeline. So I took this specific use case to give you an example. So let's imagine that we are a breeder, we follow this breeding scheme, and we want to predict our best performing lines based on their phenotypic, genetic, and environmental data we gathered from multiple heterogeneous data sources. So the question is, how can I do it easily, rapidly, and at the same time being sure I didn't throw away any potential promising line. And so the answer of this complex issue is machine learning. So at Dorian, thanks to our researchers, we are developing complex algorithms based on big historical database in order to anticipate your decisions based on your multiple previous actions and make you gain a significant time. It came the time to conclude my presentation. So I focus today on showing you standard and handy tools to use statistics in your breeding process to support your choices and give accuracy to your results. Indeed, statistics are a key point for studies validation and for confidence to the results. Statistics respect the rules of the 3 C's that is essential in the plant research world: communication, credibility, and convergence. All along the breeding workflow, from the parents crossing to field trials, statistics are essential to perform all these steps and make the best decision for parental choices, material selection, or treatment performances. So along this webinar, based on Dorian experience, supporting breeding organizations along their project of R&D data management, we highlighted 5 statistical tools available on the R&D experience software that could make the difference: efficient GxE Matrix, experimental designs with automatic material randomization, detailed ANOVA with mean comparison, GxE Matrix for better variety selection, and finally interactive graphics to gain time. I can also add artificial intelligence. But 30 minutes is too short to discuss all the possible statistic tools that can be used on R&D experience. And you can guess this list is not exhaustive. Many other tools are integrated in the software to assist you and make you enjoy statistics in your daily work. So I thank you all for attending the webinar, and I will take a few minutes to answer general questions. And please don't hesitate to scan the QR code on the screen to book a demo with us and have an overview of the solution we offer. So thank you very much.